Welcome everyone to this course on molecular diagnostics of water for microbial quality monitoring. Right. So we have around 18 participants. So a lot of you are from Jammu itself, but I think only three people are from IIT Jammu, and all of you are PhD students. So thank you very much for participating. I hope you gain a lot of information which you can use in your PhD work. Several of you are, two of you are integrated masters student from University of Rajasthan, Ajmer, right? And two of you are from SCOST Jammu. One of you are a PhD from SCOST Jammu, and then rest are bachelors in biotechnology from PSPS Government PG College from women, right? And then there's one student, Rajat Sharma, who has come from Tindola, Uttar Pradesh, and he is from Sri Ram Swaroop Memorial University, Institute of Biosciences and Technology, and he is also doing his bachelor's in biotechnology over there. So we have more of us are bachelor students. Some are few. I think only two are master students, and rest all are PhD students. Right. So this is a good mixture. I think the work we do it's very high quality work. This workshop is very rare. I think for the first time we are doing it is in India. Yeah. So this is happening for the first time in India, and this so Professor David Warner, he's a professor in Newcastle University. He has a very diverse background, and I think in the brochure I have already given his entire description and biography. But one thing very important is that he's such a humble personality and very down to earth and so approachable. And this really, when I was doing my PhD and postdoc in Newcastle University, and my PhD project was interdisciplinary in nature. It was related to genomic computation, computational genomics. So at that time, I was talking to my supervisor Rishad, and I told that this is all right what we are doing, data processing, but then who is the end user? Who will use this entire algorithm hardware approach, the processing or computational solution which we are giving? Who will be using it? So then, surprisingly, one day there was a poster presentation for all PhD students, and I also presented a poster over there. And Professor David was there, and then Rishad introduced me to him. And then the conversation started at least. That was around 1.5 years before I completed my PhD, 2 years around. Then COVID came by, so we could not proceed further. But then eventually, I really wanted to take my PhD research somewhere to convert. See, now the world has changed. It's not just limited to research papers. It is also about taking your innovations to to ground level where somebody will be using it rather than this being on the shelf your entire life. So with this objective, I wanted to find an application where we can use my work, and so I started approaching Professor David, and we also got some small funding from the Newcastle University, and then fortunately things got started. I got an opportunity to work in the same workshop which was held in Newcastle University in I think 2021, if I'm not wrong. If I think 2020 or 2021. So yeah, 2021 actually. So then, following that, I really like this workshop and the innovations which Professor David's group has done, along with his PhD students. They have, for the first time in this world, they have developed a suitcase laboratory. So for doing monitor diagnostics on site at any location, so they just need the, a, a suitcase which will have all the equipment necessary to do this molecular diagnostics work. The only thing which was required was shade and electricity supply, and for that they got a laboratory on wheel. So they have a van which was equipped with other stuff as well, including some refrigeration facility and things like that. So that was through that they were able to get the shade and electricity. And we actually during this workshop on the day three, we went to some on-site location and we got the sample collection. We collected the sample, we measured our temperature and other physical parameters, and then finally we practiced genome sequencing on some extra flow cell. So and then finally we obtained the data of different water samples collected around Newcastle from Tyne River, the tributaries of Tyne River, especially the Ouswan River. And then finally, we analyzed that data. And in the last day, day five of that workshop, we had symposium. So we'll be kind of following a very similar trend. And he has organized this workshop around the globe in Ethiopia, Addis Ababa, in Brazil, in Thailand, in Malaysia, I think, and several other countries. I may not know, but he has been traveling across and training people how to do this, how to make help us make our, or at least gauge how good our environment is, how safe it is to use. uh, Even in the UK, he found some, very astonishing results. What happens, and this microbial diversity changes through season to season. So in summers, it's something else, and it depends on country to country as well. So just yesterday, we were having this conversation that in Ethiopia, where it's dry weather, tropical weather. So when it rains, then the microbial concentration reduces because everything gets diluted. But on the other hand, it's completely opposite in the UK, where when it rains too often, and what happens is that the sewages are not able to hold that pressure, and they have to release some of untreated water into the streams as well. So then the microbial diversity or the microbial concentration increases in the water, and that may be proportionally related to pathogenic concentration as well. So it depends on season to season, region to region, and India being a very diverse country, we have very little information of what our water holds in this country. So with this vision, I when I joined, I actually wanted to start working this, and it took me around 2 years to get in here, get the funding, and it has been really difficult. And I would like to thank my PhD student Mohit and Hardik, especially. Hardik has been working with me as an intern student for last 11 months, and he has worked really hard. So all what you are seeing today has been possible because of him. Essentially, he and Divyanshi, they have done all these experiments since last 3 months. They have started slowly and slowly. We have come to this stage where we know what actually we are doing. It is a difficult work to, honestly speaking. Like when I wrote this proposal for Gyan, I actually did not know that it is going to be this difficult. So, following all these things, and we have molecular diagnostics laboratory under construction as well. So we have a container in one of the locations in the inside IIT where we have almost purchased a lot of equipments, and we will be starting our work. So this is a kickstart, and thanks to the Ubbat, you are Ubbat, right? And who's Ubbat is not here. Just message maybe Akhil and Ubbat as well to come here. So there are other faculty members in IIT Jammu, including Dr. Pratik, Dr. Ajit, and Dharitri, Dr. Dharitri. So there are several faculty members who work on, and of course BSD, Dr. Vinay. So they are very similar, especially Pratik. He also works in water environment, but they are not looking at it from a molecular diagnostics point of view. Like nobody is doing genome sequencing and analyzing, doing bioinformatics analysis of the data obtained. So this is something which we will be doing, and eventually our objective is to take it forward and look at antimicrobial resistance, the growing drug resistance, and what is going on in this region specifically, and then in India in general. So there is growing awareness about antimicrobial resistance, but most of it is still limited to conventional microbiology-based approaches. So we want to, and genomics has, it has progressed significantly, and we want to bring the benefits and reap the benefits of genome analysis uh, for antimicrobial resistance monitoring and surveillance. So with this objective, I would like to initiate and introduce this particular workshop. So what I will do is maybe I will call each of you one by one, and you can introduce yourself about a little bit about yourself. So first, I would like to call Astha Gupta from Central University of Rajasthan. Not here. So can you maybe tell Hardik to inform them they should be here, right? So because now we'll be going for sample collection as well, so they have to be there, right? So Ronak Preet, Tom, maybe you can just sign me out.
Good morning. I'm Ronak from Jammu. I'm basically from Kashmir, but I'm recently pursuing my BSc in biotechnology from Jammu, from PSPS it's called Gandhinagar, which is under Cluster University of Jammu. And thank you so much.
No, so you can also say that how are you interested in like what are
But actually, I was recently pursuing my bachelor's, and the next thing I want to do is to do my master's in microbiology.
so the next so Marvi Khajuria, she's coming. So Jaspreet Kaur, please. You can sign and then please.
Good morning, everybody. I'm Jaspreet Kaur from DCW Gandhinagar. I'm currently pursuing my BSc in biotechnology. My interest was that biotechnology have great career paths. Basically, I'm from science background. That's why I have keen interest in bio subjects. Like it's offers it's like in COVID, biotechnology performed a great role by producing different vaccines and all that. That's why I have great interest in that. Thank you.
Uh, so Anisha,
My PhD student, first year PhD student in the department of IIT. So we basically do the computational biology, biophysics related to this while we the gene sequences, particularly NGS. That's why I'm interested in this workshop because of the gene. Thank
right. So Navjot Kaur, not here. So is she coming? Why? Okay. So she will not attend the workshop at all. That's very unfortunate. So Arushi Manda.
you. Good morning, everyone. My name is Arushi, and I'm pursuing my bachelor's in biotechnology from PSPS Government College of Women Canada. Since my um, since my subject is biotechnology, and recently my subject was molecular biology, and it's very important to know about basic concepts and experiments that are being performed there. So I think this workshop will help me a lot in understanding all of those concepts.
Suzanne.
Morning, everybody. My name is Sudhir Bhatt. I'm pursuing the Bachelor's of Biotechnology in Gandhinagar, Women's Gandhinagar College. And this course basically is about microbiology, which really fascinates me about, and I'm including it in masters as well. So it basically might help me a lot. Thank you.
Right. So Simran Malhotra.
Morning, everyone. Good morning, everyone. Hello, everyone. Myself, Simran Mudra. I'm currently pursuing my BSc from BSPS Government College, Gandhinagar. I have a keen interest in microbiology, and today I'm running this workshop because it can clear my basics and can also help in my masters as well. Thank you.
Right. So, Divyanshi,
Good morning, everyone. Myself Divyanshi Gupta. I am pursuing BTech Biotechnology from Coast Jammu, and now I am doing a research intern project under Dr. Siddharth Maheshwari at IIT Jammu. I am very interested in this workshop because of the microbiological work, as it is very fascinating, and also because of the genome sequencing, as it will be very good to learn it in the early year of my graduation. So, thank you.
so Apeksha,
Good morning, everyone. I'm Apeksha, currently pursuing Bachelor's in Biotechnology from PSPS GCW Gandhinagar. So, in our current semester, we are having more bio as one of our major subjects. So, that really prompted and emphasized me to join a workshop like that, having relevant background. Thank you.
Diksha, Javra.
Hello everyone, myself Diksha Dhundya. I'm currently pursuing BSc Biotechnology from BTM Science College. Actually, I have always been excited and fantasized about the technologies and processes used in the biotechnology lab. And our lab doesn't have that facility, so I'm really excited.
It is, I mean, from my experience also, getting conventional microbiology lab also up and running has been really challenging. So I can understand there is some difficulty in those acquiring those facilities. So, Akhil, so please sign and introduce yourself.
Hi, my name is Akhil Gupta, and I am pursuing PhD from IIT Jammu. I served for 2 years as a project officer here in a water quality testing lab. And my project is on valorization, potential valorization from waste sources to value-added products. And in this context, I'm trying to isolate the bacteria from wastewater to potentially produce a high value product. So I'm looking forward for the enhancing the knowledge from this section. Thank you so much.
Palak,
Good morning, everyone. My name is Palak Manhas, and I am currently pursuing my bachelor's degree from the Padma Shri Padma Sachdeva Government Women College, Gandhinagar. My purpose for attending this workshop is to get a better lab experience that we don't get in our college, and I hope I will get that lab experience.
Ubbat, Akhil, can you call Ubbat? 11? No, no, but it is important to attend. Uh, so, no, no, so she mailed regarding that she has to give some small session on that, right? So, right now, tell her to attend. We will deal with that later. Arisha Gupta, Arisha, she is not here. So, Acha, Arisha is also from Central University. So, actually, the two girls coming from Rajasthan, so their train got cancelled. So, most likely they will come tomorrow morning. Rajit, please.
Morning, everyone. My name is Rajat Sharma, and I'm currently pursuing BTech Biotechnology from SRME. I do not have such great experience, but I have written two review papers related to on oncology and neurotransmitters. And my major interest in bioinformatics, how we can use the bioinformatics in meta-analysis. I'm currently doing this thing in under my professors and my faculties, and I'm very eager to know how meta analysis work on such things. And I wanted to do internships on bioinformatics. Currently, I'm enrolled in ISO Bhopal for the internship of bioinformatics. How the basics of bioinformatics, how can we use, how we can learn the things, how can we apply the things, and how can we use the software to sequencing the data, and how we can use the DNA genomes to analyze the whole functions, and wanted to know everything. But yes, I have less experience, as I think that I am very new for here, and I want to explore everything. And I love to do this workshop because it's contain very interesting parts like genomic sequencing and bioinformatics, parts of some bioinformatics. Thank you.
Right. So I hope that your experiences are great here and you get to learn a lot. So what we can do is then maybe I can hand over the dice to Professor David and can start his session. So do you want the HDMI cable to be attached? Okay, so just meanwhile, we are getting set up. Maybe I can ask Marvi to introduce herself as well. Marvi, can you please?
A very joyful good morning to everyone present here. Thank you, sir, for selecting me in this workshop. My name is Marvi Kujuria, and I'm a PhD scholar from SCOS Jammu. I'm not having any molecular background, but I am having a background in entomology. But my work is regarding pesticides remediation and heavy metals, so that is why I was interested in this workshop. Regarding molecular tools, many tools are there, but to identify a microorganism, all the molecular tools like PCR, genome sequencing. So that is why I have joined this workshop. So thank you so much, sir, for this honor.
A very good morning to everyone. My name is Hardik Gupta. I've been working here since 10th month with Dr. Siddharth. Firstly, I started with AMR research on basically on bioinformatics. Mostly, I've been dry lab in like in my BTech, but currently from 3 months, I have like shift to wet lab. The experience is overwhelming. It's pretty hard, the wet lab thing. But yeah, it's cool. So I just want to work on molecular things. I want to develop some pipelines related to AMR. So I want to retrieve my own data related to AMR. So maybe I'll pursue my PhD in this. That's why I've joined the work. So thank you everyone.
Now frame any of those slides. It's what we do. No, so what I can do is I can maybe the link. the online job, particularly.
Hey, good morning, everyone. Good to hear you all introduce yourself in English. I hope you can understand me well. I'm not English. I'm teaching in Newcastle, but I'm Swiss. I'm from Switzerland. I have an accent, yeah. But I hope you can still understand what I'm saying. I'll try and speak slowly. Uh, Welcome. I'm very happy that you show an interest in this workshop. We've done this in different places around the world because we think these are methods that people should know about because they will be very, very important in your career if you want to work in biotechnology, environmental science, in water treatment. We think these are the technologies of the future. So in the wrong slide, so here. So like as we mentioned before, our idea was that we can take our lab our laboratory and put it in a suitcase. If we can do it, we can go anywhere and do the testing. So we are not reliant on local facilities. And we were interested in different ways of looking at bacteria, especially. So one method that you've been talking about is called sequencing. So you read genomic material, if you can read it, that's all the information about how biology works. Very powerful technique. Then maybe from COVID, you're familiar with a different technique. It's called quantitative PCR. So if you do a PCR test to see if you have COVID, this is the method that you would be using in the laboratory to do the testing. And finally, the old school, the way we've always been doing it, is still very important, is culturing the bacteria, and then you can investigate the bacteria. Switching so quickly. But so this is the suitcase laboratory, and the main point I want to show you is that all the instruments are very small. You actually need a lot of different instruments. You need pumps, you need filtration units, you need things to extract the DNA, you need agitation vortex, you need a centrifuge, and then you need to quantify the DNA, and you need to do qPCR, which is this instrument, and this instrument is for sequencing. So the whole point is, nowadays, with the rapid progress in technology, these pieces of equipment have become very small, and also they're not that expensive anymore. So I think in 10 years, maybe everybody can kind of buy it and do the testing themselves, maybe on the smartphone. So I think that's some sort of
Oh, it's the-
Let's hope this doesn't move as quickly anymore. So this is the comparison, the conventional equipment. This is what you have in a typical laboratory. And you can see for sequencing, it costs you about £100,000 if you want to buy Illumina, and it's 60 kilograms, so very heavy. And then for qPCR, costs you £15,000. The instrument is 21 kilogram heavy. And the versions that we have in the suitcase laboratory is sequencing devices from Oxford Nanopore. So it costs only £1,000 and it's this big. And so it's very easy to take with you in a suitcase. So in terms of the changing costs, you have a 97% reduction in the weight and you have about 99% reduction in cost. And I think this will enable people that don't have a lot of money, that don't have very big laboratories to still use these technologies. I think it's very exciting. We also think that these skills that you need to do the analysis, you can actually learn them. So we work with people in Africa, these are also master students. They can do a workshop like this and they can learn the skills. So you don't need someone else to do it for you, you can do it yourself. So just to go through very briefly some of the basic concepts, but maybe you know about these things, but just to repeat them again. So DNA, deoxyribonucleic acid, it's double-stranded and it contains all the information about how something that's biological, how it works. So the information about me is in my DNA. All the information about how my body operates. So this means that when we can read, if we read the DNA, we know everything about the organism. And that's the power behind DNA or genomic methods. So the basic block of the DNA, the building block, are these nucleotides. And the important thing is there's a base. So these bases, the sequence of bases in your DNA determines the information. That's the code in which the information is stored in the DNA. And then it's kind of the backbone makes the structure of the DNA. So one gene we are interested in this workshop is the 16S rRNA gene. Have you come across this? Why are we interested in this gene? All bacteria have it, yeah, because it's very important. It's for it codes for the protein that produces the RNA. Everybody needs RNA. So every bacterium has this gene, but there are some small differences in the gene sequence for different bacteria. So if we can read this gene, we can see which bacterium it is. Do we have a 16S gene? Humans. What do we have? We have 18S. So if we want to study humans, we need to study 18S gene. If we study bacteria, we study 16S gene. So this just allows us to say if it's Escherichia coli or Vibrio cholerae or Bacillus, I don't know what. If we can read this gene, we should be able to get that information. So as you can see, the length of the gene is about 1,500 base pairs. So what we will try and do is amplify, multiply the whole gene. So our amplicons, the product of our PCR reaction should be 1,500 base pairs. And then we should read all of these base pairs. And if we do this, we can use the information to identify the bacteria in the sample. And it allows you to see within a water sample, you have so many bacteria, you have millions of different bacteria. So with this technique, you can identify what sort of bacteria you have in your sample. It's just one measurement. You can see all of the bacteria in your sample and how many of each kind. So that's kind of the powerful next generation sequencing method. The other thing we will look at or we will use is PCR, polymerase chain reaction. Have you studied this method? Have you learned about it? Yeah? I always say it's like a photocopier. You just copy your gene. Yeah? So this is the gene, and then what we have some primers. They attach either side of the gene. So what we need to do is, this is double-stranded, this DNA, so we separate, and then we attach the primer, we synthesize, we replicate, and then we can do the reaction again. Yeah, so that's the PCR reaction. So these steps are called separating it. It's called denaturation. We do at 90 to 96 degrees. And then we cool it down to 68 degrees for the primers to attach, annealing. Yeah? And then synthesis, elongation, we do at 72 degrees. Then we start again, like the synthesis. And hopefully it will work. qPCR is the same, except we can observe because we have a dye in the system. So the more copies of the gene we have, the higher the fluorescence, and that allows us to quantify the number of genes. So that's quantitative PCR. So for sequencing, we use a method, it's called nanopore sequencing. So this nanopore is like a protein with a channel in the middle. And the DNA strand will be pulled through that pore. And as it goes through the pore, it's embedded in a membrane. The currency in the membrane will change. So there's an electricity output. And depending on what's inside the pore, it will be slightly different. So this is how we can identify the sequence of the bases in the DNA. So maybe the classic old method, Illumina, is has some advantages and disadvantages. So the good thing about Illumina, if you read a short section of the gene, it will be very accurate. It will tell you exactly which bases are in that sequence. That's the advantage. The disadvantage is you can only read a short segment. So if you look at the 16S gene, you will normally just look at this proportion of the gene because it cannot do very long strands. Whereas this nanopore approach, you can do as long a gene as you find in the sample. So there's no limit. The limit is how you process the sample. If you mix it too much, you break up the DNA. But for the technology, there's no limit. You can read for as long as you want, as long as it's attached to one strand. The disadvantage is interpretation of the signal is difficult, and we sometimes don't get the right basis. So we say the accuracy is lower. So I just want to go very briefly through the workflow, and this is what we're going to do this week. We go through this workflow. So first of all, you need to take your sample. Yeah? And when you take sample, what's important is you shouldn't contaminate it because you have bacteria on your skin. You don't want these bacteria or flaking off your face. You don't want this to get into the sample. Also, if you use wastewater sample, you don't want to contaminate yourself with the wastewater. Once you have the sample, you need to concentrate the biomass. So we don't want the water, we want the bacteria in the water. So we need to separate water from the biomass. And normally we do by filtration, or we can also use centrifugation. So this we will do tomorrow morning. Yeah. Then when you have the cells, the bacteria, you need to break them open so that the DNA comes out. We call cell lysis. So that's nucleic acid extraction is the next step. And then you want to get rid of everything that's not DNA. We call cleanup. So everything else is just going to produce problems for you. When we have done this, we can then work with the DNA. So we will quantify it, and then we can prepare the DNA for sequencing. That means we need to attach some things. Well, first of all, we need to isolate the 6S gene, and then we need to attach some adapters either end of the DNA so we can pass it through the nanopore. Then we can do the sequencing. Hopefully we will be successful. We will see. Once you have this information, you need to interpret it. This is where bioinformatics comes into play. So you need to somehow translate the output of the instrument, which is a current signal, into the base sequence. We call this base calling. And then once you have base calling, you need to match your sequence to the database. We call taxonomy or biological interpretation. And then finally, once you have your bacteria, you need to interpret your data. How is the wastewater different from clean water? And this is done with multivariate data analysis. So these are the things we're going to do in this week, step by step. And I think by doing it, you will understand it much better than if you just see a lecture like this. I want to also very briefly talk about some of the hazards. So the most important thing is that at the end of the workshop, we are all going home and nobody has been injured or ill from the activities. So if you work with wastewater, it's possible because some people are sick and then the pathogens, COVID, for example, is shed into the wastewater. So we need to make sure we don't get infected from potential pathogens that could cause diarrhea, for example. Also, some of the chemicals we use, they can irritate your skin, or if you get them into your eyes, they would irritate your eyes. So we need, especially for the DNA extraction. So when we do this, we should wear the gloves to protect the skin, and also goggles if we do some of these extraction steps. And then it could be that the equipment has some fault, and we need to make sure that that doesn't happen. So that before we use the equipment, we should look at it, make sure it's in good working order, and we should follow the instructions from the manufacturer. And then, the most common accident is people tripping over something or falling down. So also we need to make sure that when you work, you're kind of aware of your environment. If there's no cables on the floor that you will trip upon. So the way we can control these risks is by thinking about exposure pathways and how we can avoid these exposures. So for wastewater, I think the risk might be if you have it on your hands and then you don't wash your hands, you're eating something, then these bacteria can go from the wastewater to your skin into your mouth. Yeah? So we're not going to eat or drink while we do the laboratory work. Before you eat or drink anything, you should wash your hands with soap. Yeah? Also, you take the gloves off at the end of the practical. The important one is your mobile phones. You shouldn't touch your mobile phones with your fingers, with your contaminated with wastewater. And then for chemicals, it's mainly the risk of thermal contact. That's when you wear gloves. It kind of provides a barrier. And also, you don't want to splash into your eyes. So if you have, don't wear glasses, you should wear safety goggles. And then for instruments, for example, the centrifuge, we also always need to make sure it's balanced. You know, the same amount of wires on either side of it, so that it doesn't spin unequally. So how can we control the risk? We shouldn't need food or drink in the laboratory. We can do it outside, but not inside. We have disinfectants that we can use. And for example, when we have the wastewater sample, we'll add some liquid to it. We'll actually disinfect it. So that will help. And also we should wash our hands with soap at the end of the work before the break. And the plates that we have, we need to autoclave it to kill all the bacteria that are on the plates. For handling chemicals, we should wear gloves and goggles, and we should look at the equipment, make sure there's no visible damage, and follow the instructions. If you see anything like cables on the floor, let us know so we can make sure there's no trespass. So that's the introduction. We'll go through this all step by step, very slowly. Normally nothing can really go wrong, you know, maybe it doesn't work, but it's not like there's a very dangerous chemical reaction happening. It's not like that. Normally we're just working with water samples. So I think the important thing is if you're not sure about something, just ask first, and then we can give you instructions before you do something. Because you will see so many steps, 1 step, 2 steps, 3 steps, 4 steps, 6 steps. Any step you make a mistake, basically, then the sample maybe is not working out. Do you have any questions? No? Okay, so that's the introduction.
Last slide. This one is the valve, basically, which we have. First of all, you can wear this. Okay. It will be nice, but optional. Also, as Professor David has given us all information related to hazards, so please be very careful. So our plan is that we'll go to Surunsar and Mansar Lake. So there are some lakes nearby. And then while coming, maybe we will pick up some sample from Tabi River in Sidra. And then after coming back here, we will pick up some samples from our STP plant inside IIT Jammu. But at any stage, if you feel that there is, even a hint of danger to yourself, you are not under any obligation or you're not being forced to do that. You can just stay away. The most important thing is to safeguard yourself. Okay? Work, we'll deal with it later on, but you just be very sure that you don't, put yourself in any kind of danger, right? So I hope this is very clear, right? Even if I or anybody says they'll do this, if you think that no, this is something I don't want to do, it's perfectly all right to say no, and you don't need to do it. Okay? Don't be in any obligation. Your safety is paramount here, right? Okay, so I will now pause recording because we need to go on site. Okay, thank you.
Okay, here we are at Mansar Lake and taking a water sample in this spot for people to feed the fish.
So here is Akhil, who will be collecting sample for us. He has pH measuring strips as well, and we have brought some devices to measure temperature, pH, and also dissolved oxygen. And we have these ice boxes here. Once the sample is collected, we will keep the sample after measuring pH, temperature, and dissolved oxygen. back in the box, right? And we'll take it back to the campus and we'll use another set of instruments to again pre-measure it. PM. Monster Lake. 21st April. Coordinates we can measure later on. Yeah, so 7.5 is the pH. Right? Okay. हार्दिक थोड़ा इधर So this is for what? So Akhil is going to use this particular instrument to measure dissolved oxygen in the water. So collect it, no? Collect it in the bottle and then you can measure. So we suggest that there's too much movement in the water, so it's difficult to measure. So once I told that let us collect the water. But that is cold water. So again, वहीं से from there itself. They think that we are giving उसको So let us So what is that? अच्छा,